Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Om Swastiastu, Shalom, Namo Buddhaya, salam kebajikan dan salam sejahtera bagi kita semua. Salam agromedis. The Honorable Dr. Ulfa Elfiah, Reconstructive and Aesthetic Plastic Surgery Specialist, Subspecialist on Burn and Wound Consultant, as the Dean of Faculty of Medicine, Jember University. We also welcome all the Vice Dean, our distinguished speaker, Professor Dr. Jenny Klein Nuland. Also greetings for all working units, our treasured staffs, lecturers, and students of Faculty of Medicine, Jember University. Ladies and gentlemen, stem cell technology represents one of the most exciting and rapidly advancing frontiers in modern medicine. Its potential to revolutionize treatment strategies, particularly for musculoskeletal defects, opens up new pathway for healing and recovery. Recognizing the importance of staying ahead in this dynamic field, the Faculty of Medicine, Jember University, is honored to present this public lecture, Stem Cell Technology for Musculoskeletal Defects. Allow me to introduce myself. My name is Gessi Oktanabila, the host who will guide this event until finished. Moving on to the next agenda, the Indonesian National Anthem, Indonesia Raya, Jember University Hymn, and the Mars Agromedis will be played in a minute. I kindly ask all attendees to stand up. Ladies and gentlemen, you may now have your seats again. Moving on to the next agenda, I would like to invite the Dean of the Faculty of Medicine, Jember University, Dr. Ulfa, to open this agenda and give in the speech. Please welcome Dr. Ulfa.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, esteemed faculty and students. My great pleasure to welcome you to today's guest lecture featuring Professor Zineke Klein from Amsterdam. We are honored to have her here to discuss the groundbreaking advancement in stem cell technology for addressing musculoskeletal defects. As future medical professional, understanding these innovative therapies is crucial as they hold the potential to revolutionize treatment options for patients suffering from various musculoskeletal. Professor Zineke's expertise will provide us with valuable insight into the mechanism of stem cell application and their implication for degenerative medicine. I encourage all of you to engage actively during the lecture and take advantages of this opportunity to deepen your understanding of how stem cell technology transforms patient care. Please join, give a warm welcome to her and your today's guest. Thank you.
Thank you, Dr. Ulfa, for amazing speech. Next agenda, we're gonna start with praying. To Mr. Muhammad Yassin Assas, time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Sebelum acara guest lecture dimulai, mari kita berdoa menundukkan kepala agar supaya acara ini senantiasa mendapat ridha Allah dan diberi kelancaran. Izinkan doa kami pimpin secara Islam, bagi yang beragama lain selain Islam dapat menyesuaikan. Bismillahirrahmanirrahim. Alhamdulillah rabbil alamin. Hamdan syakirin, hamdan naimin. Hamdan yuafi ni'amahu yukafi mazidah. Ya Rabbana lakal hamdu kama yanbaghi li jalalika wajhikal karim azim sultanik. Allahumma salli ala sayyidina Muhammad wa ala ali sayyidina wa maulana Muhammad. Segala puji bagi Allah, Tuhan semesta alam, yang dengan rahmatnya kami bisa berkumpul di tempat ini dalam keadaan sehat walafiat. Kami bersyukur atas nikmat iman dan kesempatan ini untuk mengikuti acara guest lecture. Ya Allah, ya Rabb, Engkau yang maha penyayang, kami mohon keberkahan dan petunjuk-Mu agar acara guest lecture ini menjadi ladang kebaikan. Bimbinglah kami dalam setiap langkah kami untuk mengambil manfaat dari ilmu yang akan disampaikan. Ya Allah, kami berharap acara guest lecture ini sukses. Bukakan pintu ilmu yang bermanfaat di hadapan kami dan berikan pemahaman yang mendalam kepada kami. Semoga ilmu yang kami dapatkan menginspirasi kami untuk berbuat baik dan menjadi teladan di tengah masyarakat. Ya Allah, satukan hati kami dalam kebaikan. Permudahkanlah kerjasama dan kebersamaan di antara kami selama acara guest lecture ini berlangsung. Bantulah kami untuk saling mendukung dan menguatkan dalam membangun berhubungan yang harmonis dan berbagi ilmu. Ya Allah, dalam ridha-Mu kami memulai acara ini. Kami berharap agar setiap momen di acara ini memberikan manfaat yang abadi dan meningkatkan kebaikan. Terima kasih atas segala nikmat-Mu dan Semoga acara ini menjadi ladang keberkahan bagi kami semua. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina adzabannar walhamdulillahirabbil alamin. Assalamualaikum warahmatullahi wabarakatuh.
Our distinguished attendees, before we start our lecture, I kindly ask the Dean and the Vice Dean, our special guest, Professor Janike Newland, and the doctor who accompany her, Dr. Rasita, to come to the front because we're going to take a group picture together. Hold a second, Doc. Yes. One more time with the audience, please. For all audience, please stand up and come to the center so we can take a picture of you. Thank you, Doctor, and all audience. Thank you for your willingness to take a photo together. I would also like to extend my gratitude to Dr. Ulfa for taking the time to be present in this event. We would be honored if you could stay with us until the event concludes, but if you have another commitments, please feel free to depart. Ladies and gentlemen, now our guest lecture will be start. This guest lecture will be moderated by Dr. Laksmi Indreswari. She is a general surgeon who now works at the surgery department at hospitals in Jember named Subandi Hospitals, Jember University. She is also a part of the Anatomy Laboratorium of Faculty of Medicine, Jember University. Please welcome Dr. Laksmi.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody. My honorable dean, my dearest lecturer friends, and all of my lovely students, and everybody that's joining from the online, from the Zoom. Before we start the lecture, I would like to read a few curriculum vitae. Can I please ask the curriculum? Professor Clay Nolan got pages of curriculum vitae. That's why I'm emphasize her most important work, because she's professor, academic center for diversity. One thing that she's proud of work visiting, especially in the Philippines and China. And she also works on products from Karagina. Of course, work about STEM. Which and its function is repair bone regeneration or inhibit tumor growth at 9 works and joining in this because ending. So with no further ado, please welcome Professor.
Thank you very much. I will talk to you stem cell technology, musculoskeletal basic. Amsterdam, where I'm from. Hey. Goes in Amsterdam Canal. Beautiful. See the typical canal in the Amsterdam slide. The ACTA building, and that's the academic center of Amsterdam. This is the building, Dutch, it's Academic Centrum Tandheelkunde. The bottom clinical part where we have a research tower. Actually, dental faculty, special laboratories, that part thing. Excellent. Now, for research, which we are doing, is tower. All this research is collaboration. Say that, but here we see the left side a natural tooth, and on the right side the implant. The major difference between natural tooth and an implant, around the tooth is periodontal. So, but anyway, like to left periodontal ligament, that is soft tissue between tooth and bone. This little therefore, again, it's about the periodontal ligament between the bone is yellow, the tooth orange. There is soft tissue periodontal. Oh, if it's lost, many patients. It's titanium. It's directly bone. It's no between. That's a major. What is implant dentistry as well as orthopedics? That is direct attachment bone to the implant. We call that direct, no soft tissue. Here we have a problem. How do we solve this problem where there is a lack of bone? Here we are looking at teeth, and you can clearly see the implant. The implant of a tooth is going into the jaw bone. Why can we see implant? Because all the bone is implant, so that implant will also be lost. That's the problem then for the patient. Questions here. What determines exactly implant stability? Done, there's insufficient bone to place an implant. Here is typically no bone to place the implant. Which factors can stimulate bone healing? That's a very important question. Bleeding or bad. And what causes clinical picture? The point is clinical problem. Problem is bone loss. Bone loss is there not good osteointegration, whereby bone implants. What is definition is direct between the living bone, load-bearing implant without, in short, direct between. In order to understand how that works, or what is so conduction, induction. And here we see that osteoconductive permits bone growth. So osteoconductive means bone is growing surface, for example, of an implant. Osteoinduction is something else. Means that material that it can resemble cells. It can lead to transformation on different mature bone forming, and it bone formation topically will put their bone in production of topic bone formation, planting, induction, stem to become, in this case, to become bone cell. Which factors do? That's bone properties. The bone properties that are important for plantation, bone density, activity of the bone cells. The bone cells, there are different types of bone cells. The blasts, osteoclasts, and I will get back later to implant properties. Very important, as the surface microtopography. It's a difficult word for the roughness of the plant. Plant can be smooth. That's very important for cell attachment. And the surface chemistry is important. Application of biomaterials is important not only in dentistry, as we see on the left-hand side, where we observe titanium screw thread implant, as well as a very smooth implant. Next to it, you see from the orthopedic, from orthopedics, an example where is a big bone defect in the leg. The bone defect is filled up with I think a 3D printed material. Crucial is that this material is also integrated bone. Not only for that type of orthopedic, but also on the right is visible a hip implant. Most important is stability implant. Implant has to be stable in the bone, not be in the of the implant. It's a big problem for the patient. What are now methods to improve osseointegration? We like to improve. The better the higher the survival rate of. So we like to improve lesions per pre-osteotic stem cells. We want more adhesion. We can coat with extracellular, because the stem cells. We can also enhance proliferation, which is cell. And we can do that by coating with calcium as a growth factor operated. This is an important modern way. BMP2, bone morphic protein 2, osteoinductive stimulus to become only bone cells. Stem cells can be different types of bone cells. That's important around the implant. We can promote mineralization of bone matrix by using fluoride, regulate bone remodeling by inhibiting the bone resorbing by using. Whether you are familiar with fluoride to promote this whole work to prevent action. If we inhibit bone resorption, prove so into. So we did a study. This is a little bit difficult, but I will try to explain it because it's a new method we developed to stimulate integration, stimulate cell attachment to an. This method is called RGD functional supported lipid bilayer or differential. What it means, I will explain. Biomaterials for bone mentioned, important in dentistry, orthopedic. Cells play a crucial role because they need to attach. What are currently doing something? We take biomaterial that is coated with RGD. I will get back to that, what it means. Because RGD cells love RGD sequence to attach. But if normally the biomaterial is stiff, we have thought about something that we made supported lipid bilayer. That is 2 layers of lipid move a little bit, like in your cell membrane. So it's movable. That supported lipid bilayer, RGD sequence, that is the part of the talk. To invent cell morphology, shape of the cell, and the osteogenic potential. Can they make bone? Then there are RGD supported lipid bilayer. So what we see in the pictures is that adhesion and the morphology change, perhaps, of supported lipid bilayer. I want to say the morphology dictates its function. So if it changes morphology, it will change function. Finally, the osteogenic potential, probably enhanced, that the stem cell becomes osteoblast, will make bone. So we were thinking that it was a wonderful make this supported lipid bilayer compound we call PC. That compound is important in making the layer. Here we see these round circles. If they attach to glass, go open, stretch up, shown in the picture. Then lipid membrane, cholesterol, where RGD lipid bilayer in the membrane. If you eat a lot of fat, you have cholesterol going in your membrane. Cholesterol goes easy in the membrane. What we did is start cholesterol. And then we measured how they adhere. Morphology, we measured their differentiation. Why is it now again so important? RGD sequence. Because cells love to adhere to RGD. They have. We look in yellow, nucleus. And the blue and green are the RGD integrin sites bind to the RGD in the intracellular matrix. Normally, in extracellular matrix are molecules such as prolactin, pontin, arginine, their molecule. Cells can attach with the integrins to the RGD. We wanted to improve osteointegration. We used lipid bilayer. Lipid bilayer is present, as shown here, also normally in the cell membrane. We put it on a glass substrate, then the lipid membrane, which is movable. We stick in there the cholesterol and RGD, measure the adhesion. We are looking here at 5 pictures. The left polylysine is, let's say, a control. SLB is the supported lipid bilayer. And then we have 3 pictures of supported lipid bilayer concentration. We do see that cells, no difference observed. We quantified the cell density. Cells the control is still doing best. Most cells still attach to the control PLL, and they maybe a little bit lower on supported lipid bilayer. But we were developing a new method for stem cell to this. We also looked at the morphology. The morphology changes a bit. If you look on the polylysine, the cells are more round. If we look at other picture, clearly see the cells become more elongated or longer. So we quantify the surface area of cells on supported lipid bilayers less than control. On the other hand, the eccentricity measure of being long or lower eccentricity, see that be first higher than that of inclusion. Cells are more round in our control and elongated. Supported lipid bilayer, so more elongated, higher concentration. So something is happening. We also osteogenic cells, when seeded onto these substrate, gene expression of osteogenic RUNX2, osteopontin, collagen, alkaline phosphate. To make the long story short, after 17 hours and a week expression, osteopontin and collagen were significantly changed when the cells were cultured on the supported lipid bilayer. The other genes were gene expression was affected when we did culture lipid bilayer. So to summarize, and what can we now do with this information? Our cultured movable fluid like RGD functional supported lipid bilayer. It was shown that it modulated the adhesion of the cells and it enhanced the differentiation. What can we now do more? The lipid bilayer is a little bit fluid. We can make it more fluid, less fluid. And that might be different stem cells to attack. So that's our future work, to know better what are the details here and improved attachment. What else can we do? We can, now we only with our, maybe what we can do is we insert cholesterol, something else of it. For example, one of the compounds enhance bone formation by stimulating the stem cells to become bone. So we concluded that supported lipid bilayer is a very promising substrate to develop innovative coating for biomaterial, bone regeneration, both dental and orthopedic. This is then about how we can improve from a biological point, technical point, bone adaptation. The study I showed you, it was collaboration with Dental Unit, it's a technical unit. They developed bilayer. We as biology, we culture the stem cells on beneficial. Now, what now about bone adaptation? Then from the dental, it's clear that bone is strong. In dentistry, bone has to carry the jaw chewing forces. Those are very high. Orthopedics, you have long bones to carry weight. Bones have to be strong, have to adapt. They would break. bones micro. I wish that you remember bone contains many blood vessels. Blood vessels are crucial for bone, otherwise the cells will die. Around the blood vessels in the hard bone matrix are osteocytes, the bone cell. Go to the. See them again. The scheme is, let's say, part of the big pores in bone containing blood vessels as nerves, and then on the right you have a cross section through that bone, and all the black dots are the osteocytes cell bodies. See all the black stripes that are processed. One cell, about 60 extensions, making a network in bone. This is in fact important for osteocytes form a network throughout all our bone. So bone is not dead, bone is very much alive, and osteocytes. These are two magnifications of osteocytes, showing again all their cellular. 95% of all bone cells is osteocyte. Only 5%, so not much, is an osteoblast, or that is the cells that produce bone. Most of them are. In the past, we did not know what these cells were doing in the bone, tension, and now we know that they are the mechanosensors of bone, and they are orchestrating during bone remodeling the activity of the bone forming cells, the bone resorption. So they are playing a very important role. They have a very important function. This is the cartoon of osteocytes in the center, green and the red light. And they can signal towards the osteoblasts, the OB cells, to make new bone. They can also signal to the osteoclasts, the bone resorbing cells, to resorb the bone. So these cells have a very important, and they are producing molecules that will inhibit or stimulate the bone maker, the bone. At defective mechanotransduction, the bone was not adapting to the loading anymore. So that was the proof that osteocytes are crucial cells for bone adaptation. So, and the structure of bone is mechanically meaningful. Here we are looking at a long bone and bone bearing capacity. On the left we see a tennis player. This player bones playing much that is not used for tennis playing. So this arm then the force case an astronaut space is under total disuse condition. An astronaut space loses much bone. That is a problem. It's a problem also if you want to send them to. We better know how to prevent this bone loss, because they will only gain if they come back on Earth. They will regain. So that is an example of unwanted bone loss as a result of not using bone. Example is for all teeth are extracted, then there is no loading on jawbone. On the left of these twin sisters, the feature. That's nice if there are no teeth and it's all not mechanical. Again, the adaptation of bone to mechanical loading occurs during the process. We see a scheme bone remodeling means. It is bone resorbing activity by the osteoclast. We see three big osteoclasts. On the right, they are red. And in the scheme, you see nuclei. These cells are resorbing bone the direction of the loading. We see a layer of osteoblasts. They are making new bone. The real picture, we can see the red cells that have produced the red layer, that is the bone. Osteocytes are laying everywhere what is the bone. Bone remodeling process whereby osteoclasts absorb bone, make a little hole in the bone, behind growing a blood vessel. Osteoblasts are also produce new bone. For remodeling, eating bone by the osteoblasts, filling up this hole by the how much loading does a bone now need? The optimal is 1,000 to 2,000 micros. Microstrain is like a. If you are astronaut, have no loading, you get bone absorption. Microstrain. You can also have overloading. You can have a little bit overloading or a lot. A little bit, you get bone gain. Bone. If you have more overloading, and 20,000 falling very hard, you get bone loss. So going back now to osseointegration, bone adaptation. For an implant, these crucial that it allows optimal loading of bones implant. X-ray picture of an implant and a logical picture. Why does an implant? Is it still working? An implant has a thread because in between the screws, the local loading condition is not the same as just outside the screws. So to get good osseointegration, a dental implant, orthopedic implant, the design is also crucial. Again, mechanical adaptation occurs if we have an external load leads to a local mechanical signal osteocytes. They can feel the loading because there is some fluid going over them and they deform. Then they produce a signaling model of the osteoclast, the osteoblast bone, settle together, bone mass, and so again, it's produced, for example, nitric oxide and prostaglandins that affect both cell types. I want to mention these molecules because prostaglandins and nitric oxide to block production of molecules. Bone and so hope that you know what prostaglandins are. Prostaglandins molecules take head fever, take NSAIDs, non-steroidal drugs to inhibit prostaglandin release. But in bone adaptation, prostaglandins are produced but at low concentration, low concentration beneficial for bone. Are different concentration towards. I am now coming to to explain this scheme where loading bone is low. All the open white space where you see two osteoclasts, that's resorb bone, production. What occurs in bone if it's loaded in the direction? Plasts are going in the direction where you see black locally no loading, go into apoptosis. Osteoclasts are attracted, and osteoclasts are coming from the bloodstream and create your bone in the direction where light. If we continue, we see that the yellow osteoclasts are in a region with high loading, produce other molecules such as prostaglandins that recruit stem cells to the bone surface. Cells make black blue bone. What we call adaptive bone remodeling. The active, bone resorbing and bone forming, totally under the influence of the loading of bone. Direction. Come again. Now, to the clinic, because we have done a lot of basic research. Stem cells better. What can we do with the stem cells to make bone for the patient? And the example we have used here is a patient. On the left, the patient has still teeth, and on the right, the teeth have been extracted. We are looking at the upper jaw of a patient. It's the air-filled sinus. Two sinuses per jaw. If the sinus is becoming bigger, at the bottom you see the bone is extremely resorbed. This patient without any would like to have implants, but that's never possible because the bone is far too thin to place implants. So that is a clinical problem. Not enough bone to place implants. Here we are looking at it again in a schematic figure. Jaw bone volume increases, absence of loading, and the absence of mechanical loading because teeth are absent. No teeth, no loading. Bone resource. On the left, the yellow bar indicates bone height in normal situation. And see on the right, if teeth are, bone height is very low. And no implant ever be placed there. What did we do with patients? We did bone augmentation by a procedure called sinus floor elevation. It's again the air-filled sinus. On the bottom you see the blue and the red. You see the bone height. We put synthetic bone material in with stem cells in order to increase bone height to allow dental implant placement. I'm saying what we did. Normally, or the golden standard for the patient, own bone obtained from the hip. And that is the current clinical practice. So here we see again the sinus. Then it's filled up with synthetic bone material, for example with stem, or with own bone. What we did is, 6 months after the operation, we used a hollow burr in order to place the implant. But the hollow burr, we took out the tissue, studied it histologically. So I said, the problem is that autologous bone, the bone for the patient itself, is only limited available. There is donor site morbidity. That means pain, the hip. To harvest bone, especially for the patient has more pain in the hip than maybe around the. That's not danger of infection. Always discomfort the patient. The alternative is to use a bone substitute. Bone augmentation with mesenchymal stem cells can be done, and here is a cartoon of the sinus. Normally, cells grow between granules we put in there. It's indicated by the blue arrows. The cells come from the existing bone of the patient and they grow in between the granules. In our study, we isolated stem cells from adipose tissue. We added it to the granules and then we had stem cells everywhere. So that was our idea, to have cells that can make bone everywhere. We did that in a so-called one-step surgical procedure. patients come only one time to the clinic. And what we did is, going from the left, according to the left, you see a belly of a patient, a plastic surgeon who obtained via liposuction adipose tissue from there. From the adipose tissue, which is dead, stem cells were isolated and put in a so-called solution operation. Then the stem cells were feeded onto little calcium granules. If we go to the clinical pictures, they were placed in the sinus. In the middle figure, you can see a little bit, if you look well, the like sugar-like granules in the sinus, and then the wound was closed. 6 months later, we took a biopsy because the implant is placed, and we did histomorphology, CT, and structure and histology. What happened in the patient? That angiogenesis was improved. We measured. What happened is, on the left is a bone transplant only. That means only calcium phosphate. And on the right is a bone transplant if we add stem cells. If we have only the transplant, blood vessels will grow from outside to inside. That is red outer tissue. It will grow into the graft. If you add stem cells, you get blood vessels from inside out. So still blood vessel formation. Plant, but the stem cells produced in the transplant. So enhanced and more blood vessel formation, crucial for bone formation. This study was done 10 years ago, and we did a long-term follow-up. The long-term follow-up, we did an oral examination as well as X-ray examination. Also blood were done, and we found, I will make a story. It was the same in the patients without and with the stem cells, but stem cells have accelerated bone formation. Earlier an implant, importantly, stem cells, adipose tissue, patients years later still fine. And here in this last slide is actually the cover of a PhD thesis of Vivian Wu. Did the clinical studies on this jawbone regeneration. Vivian concluded that for bone tissue engineering, what we actually patient, that growing of bone by